Okay, another. Any questions about the light reactions of photosynthesis? One more thing I'm going to mention about the light reactions. There is a second little mechanism that uses parts of photosystem 1 and parts of photosystem 2's electron transport chain. And this is a cells get switched back and forth depending on the needs. We call this mechanism cyclic, meaning circle, photo, meaning light, phosphorylation, meaning making ATP. Cyclic photophosphorylation is when a cell needs ATP only and forget the NADPH. And what we do is we use photosystem 1 and part of photosystem 2's electron transport chain. We call this process cyclic because these electrons are ejected from photosystem 1's reaction center, and ultimately, when this whole thing is over, they go right back to photosystem 1's reaction center. So let's start over here. We take photosystem 1, our reaction center here, light gets absorbed, electrons get kicked off, goes to an electron acceptor, gets passed down electron transport chain. Now what we're going to do, so far this is all what we see in photosystem 1. But what we're going to do in cyclic photophosphorylation is we're going to borrow elements from photosystem 2's electron transport chain. Specifically, we're going to pass these electrons to the cytochrome B6F complex. Now this complex is ordinarily associated with photosystem 2, but we're going to borrow it for this. We pass the electrons down here, and that ultimately is used to do active transport of H+, and we make our ATP that way, just like we did in photosystem 2. Now we're also going to borrow the rest of photosystem 2's electron transport chain. So this is associated, on the rest of this stuff is associated with PS2. Okay, so we pass it down, and ultimately they get passed right back to the reaction center of photosystem 1. Recall, photosystem 2 sent its electrons to photosystem 1's reaction center. So if we borrow this part of it, we'll send the electrons right back to photosystem 1. So the electrons go into a cycle. They get kicked off the chlorophyll, run through electron transport chains, passed right back to where they started from. And that allows us to make ATP alone, since we're not using the rest of photosystem 1's electron transport chain, we're not making it NADPH. Now, if we run low on NADPH, we can switch back to the 2 photosystem scheme. The chloroplast can switch back and forth depending on its needs. Okay, so that's... Yes? Excuse me? Oh, no. Okay, when the NADPH level gets too low. Okay. Yeah. It's very similar. It's a different complex, but it has these cytochromes, these iron-containing blood red proteins. It's just different than cytochrome C. We have one called cytochrome B6 and another called cytochrome F. Okay, but so those are very similar to these iron-rich small proteins that pick up and donate electrons.
Is that like the shuttle bus?
In a sense, yeah, the cytochrome C was small shuttle bus between the second and third enzyme. These cytochromes are actually embedded in part of this much larger complex. But those are just two of the key components in this. They gave those two moments. Okay. All right. Okay, that's our cyclic photophosphorylation. Now let's look at the dark reactions that occur in the stroma. This will go a little bit more quickly, folks. The dark reactions, folks, the dark reactions occur in the stroma of the chloroplast. And the basic idea is we're going to produce glucose from 6 carbon dioxides. That process takes a number of steps, about 7 or 8 steps, and a ton of energy-rich molecules. So the overall reaction, you take 6 carbon dioxide from the air, you got to burn through 18 ATP, and pass electrons from 12 NADPHs, and then through a series of steps you produce 1 6-carbon glucose. So 6 carbon dioxides ends up ultimately going into 1 glucose. So that's the quick and dirty of it. This is how we make glucose. This is how we make sugar. We can store the sugar for future use. We can make polysaccharides like starch and stuff like that out of it. We can convert it to other sugars like sucrose and fructose and maltose and what have you. But this is how we make sugar. Okay, and it relies on the energy-rich molecules produced in the light reactions. Now let's take a quick overview of the cycle. If you want to see every detailed step, you can go to the book on that one, but we're just going to look at the overview. We're going to start out with 6 copies Okay, 6 copies of a 5-carbon sugar. And that 5-carbon sugar is called ribulose, not ribose. Ribose goes into nucleotides. Ribulose 1,5-diphosphate. It's a 5-carbon sugar with 2 phosphates. That makes it unstable. It can be broken easily. Remember we stuck 2 phosphates on sugar in glycolysis so we could split the sugar in 2 easily? This one's already got it. So we have 6 copies of that 5-carbon sugar. Now, yes?
Isn't that RU?
RUDP is the common abbreviation. Okay. Yes, who wants to go over it? Yeah, RUDP is the common abbreviation for ribulose diphosphate. Now what we're going to do is we're going to take 6 carbon dioxides, and an enzyme will attach a carbon dioxide to each one of those 6 5-carbon sugars, the RUDP. So let's put 6 CO2 over here. Okay, now what's going to happen when we force the carbon dioxide into each one of those sugars? We split the sugar in half. We end up with 2 3-carbon fragments, each with a phosphate from each of the sugars. So we end up with 12 3-carbon molecules. No need to know what it is. Now the enzyme that does this is called ribulose diphosphate carboxylase, RUDP carboxylase. Our carboxylase is an enzyme that attaches carbons to something. This enzyme is one of the most abundant and one of the most important enzymes in the world. Because what it does is it takes the carbon dioxide and takes it out of the air and attaches it to these sugars. We often call this process CO2 fixation. We take carbon dioxide and now attach it to an organic molecule. Okay, now we got these 12 copies of these 3-carbon molecules. We do a number of manipulations to it. In one step, we're going to use 12 ATPs. And we're also going to use all 12 of our NADPHs in one individual step. So what we have to do is add phosphates here, plus add 2 electrons in each one of these 3 carbon molecules. So we're burning law of energy in one single step. We keep on playing around with this stuff, having 12 3 carbon molecules. And then at some point, at one point in the step, we're actually going to pull 2 of these things out of the cycle entirely, and another enzyme joins them together to make a glucose. When we do that step, now we're down to a mere 10 3 carbon molecules. We pulled 2 of those things off the cycle, made our glucose. Now we've got 10 copies. Question back there.
You got NADPH, losing a hydrogen.
Yeah, exactly. And losing electrons. So now we go to the oxidized NADP plus version. The H plus is released, but the electrons are passed. These are getting all these
First
here.
one up there is actually
NADPH, yeah, that's our coenzyme. Okay, we got 10 copies of our 3 carbon molecules. We're going to do a number of other steps. One more of those steps uses the remaining 6 ATPs. And then finally, we end up with our original RUDPs through a series of steps. And this is what we call the Kelvin cycle. The major means of the dark reactions. All right, so like I said, a number of steps in it, the details are in the book, but the basic outline here, 12 copies of a ribulose, or 6 copies of a ribulose diphosphate. We add the carbon dioxides from the air to that, our CO2 fixation process. We end up with 12 copies of the 3 carbon molecule. We play around with the structure a few times, use up a lot of energy, take 2 copies out of the cycle to make glucose, and then we play around with the remaining 10 copies, and eventually regenerate our original RUDP, our original 5 carbon sugars. That's why it's a cycle. Okay. All right, and that's the major means of photosynthesis. But there is a little problem here. And the problem is this. It turns out our enzyme, RUDP carboxylase. Remember when we did the enzyme lab and we said the thing we call the Km, that constant, is an indication of how tightly an enzyme binds a substrate? In other words, how well, what's the affinity of that enzyme? If you have a high affinity enzyme, one that binds substrate tightly, you've got a very low Km. If you have a low affinity enzyme, you've got a very high Km. Guess what, folks? RUDP carboxylase is terrible at binding carbon dioxide. It has a very high Km for CO2. It has a very low affinity for CO2. Now that's a problem. And one time it wasn't, because this enzyme goes back a long time ago. 2.5, maybe 3 billion years ago. In those days, the carbon dioxide levels were 50 times what they currently are. So carbon dioxide was no problem. You had plenty of it around. A lot of it would actually be almost toxic to us at those kind of levels. It would be hard to breathe in that atmosphere, even omitting the fact there wasn't any oxygen. So we got ourselves a bit of a problem here. We have a low affinity enzyme that fixes our carbon dioxide and ultimately leads to glucose production. Now, where does it run into a problem here? You have to take a look at a plant leaf. Most plants do their photosynthesis through the leaves. And I'm going to draw a cross-section of a plant leaf here. have leaves to increase the surface area for absorbing light. The more surface area you have, the better. Problem is, plants lose water through those leaves. All that surface area means on hot days, water evaporates off the leaves. They call that transpiration. And what can happen is a plant can dehydrate itself that way. One of the biggest threats plants face is dehydration. Water loss through the leaves that end up causing, have you ever seen, dry things, the plants shrivel up and their leaves turn brown, they stop photosynthesizing and they die? Okay, that's the problem they're facing. Now, most of the photosynthesis occurs in cells inside here, what we call the mesophyll. Okay, so photosynthesis occurs inside the leaf. To prevent water loss, the top layer of the leaf, the epidermis, is coated with wax. That's why a lot of leaves feel greasy on the top surface and not necessarily on the bottom surface. Because the sun's beating down on the top part of the leaves, that's where you get the most evaporation. So we coat it with wax to prevent water loss from the top. Now, water can, at the bottom, we have a lower epidermis, and there's these pores that are surrounded by what are called guard cells that can open and close. We call these pores stomata. Now the pores do this. The pores allow gas exchange. So carbon dioxide can get into the leaf where photosynthesis is occurring. The downside is through those very same stomata, water can go out of the leaves. That's where these plants are the leaves are going to lose their water, is through the stomata. So a complex system of plant hormones detect water level in the plant. If the water level is low, if the plant is dehydrated, we close the stomata. That prevents water loss, but the problem is, it means the carbon dioxide in the leaf gets quickly used up. When the carbon dioxide starts getting used up, the Kelvin cycle shuts down, no more glucose made. And the problem is that RUDP carboxylase, the one that fixes carbon dioxide, has a low affinity for carbon dioxide. It shuts down when you get the carbon dioxide level goes to about 15% of what's currently in our atmosphere. That shuts this enzyme off. So what happens is if you're in a hot, dry, arid environment, half the time you're going to have your stomata closed, you're not producing glucose, you're desperately trying to prevent dehydration. But you're not producing any more glucose. It's that balance between making glucose and losing too much water and dehydrating. But not long after the advent of angiosperms, perhaps about 50 million years or so ago, some plants, and perhaps the pioneers of it were many of the grasses. At that time, the world was getting cooler and drier, and all of a sudden you start seeing proliferations of grasses and grasslands, savanna areas and stuff like that could live in arid environments. Okay, they developed, and many other plants now that are common in dry, arid environments, developed a little trick. And we call this little trick the C4 cycle. What the C4 cycle is called that because it relies on 3 and 4 carbon molecules.
Is it high?
High affinity. The idea behind the C4 cycle is this. Instead of relying on that low affinity, low binding ribulose diphosphate carboxylase, what we're going to do is use another enzyme that can grab CO2 at very low concentrations and attach it to a 3 carbon molecule to make a 4 carbon molecule. And then a second enzyme later on will pull the carbon dioxide off that and pump it right into the Kelvin cycle. If you can do that, you can photosynthesize, or you can make glucose at very low CO2 levels, because this enzyme has a very high affinity for carbon dioxide. It grabs CO2 at extremely low levels and can attach it to our 3-carbon molecule. You have a question? Oh, okay, all right. Okay, the C4 cycle fixes carbon dioxide at high affinity. In other words, this enzyme binds CO2 really tight to a 3-carbon molecule and then ultimately releases it into the Kelvin cycle. Let's take a quick outline look at that. We're going to have a 3-carbon molecule here. We're going to take a CO2. This high affinity enzyme attaches it to make a 4-carbon molecule. We play around with it a little bit, using up an ATP in the process. Make another 4-carbon molecule. And now what we're going to do is a second enzyme remove the CO2 from that 4-carbon molecule and passes it into the Kelvin cycle. It effectively feeds the Kelvin cycle carbon dioxide when RUDP can no longer function because there's not enough carbon dioxide around. So, of course, when you do that, we go back to a 3-carbon molecule. We play around with it again, using up a second ATP. And now we come back with our original 3-carbon molecule, ready to rock and roll again. So the advantage of the C4 cycle, plants that have that, and it's only certain types of angiosperms, including most grasses and a lot of desert-type plants. Plants that have that can function with their stomata closed most of the time because it just takes tiny amounts of CO2 for this cycle to work. And they can still produce glucose. Plants without the C4 cycle, they're in trouble in arid environments. Examples of C4 plants include most grasses, most desert plants, crabgrass, especially notorious for that, kudzu, the plant that ate the southeast, that's a C4 plant too, so it can live in some pretty nasty environments and still proliferate quite nicely. So there's a number, there's a special adaptation, angiosperms only, gymnosperms and other lower vascular plants do not have this, but angiosperms, at least some of them do. An adaptation that happened tens of millions of years ago in response to a global cooling and drying of much of the global environment. And grasses, of course, are very successful plants now. Much of the world are these dry, semi-arid grassland areas. Think of the Great Plains and the equivalent in Canada, large parts of Russia, the African savannas, the Asian savannas. That's pretty widespread. Okay, so that's another little trick here. And that covers photosynthesis. And now what we're going to do in just a moment, let's take a couple minute break and then we're going to do that that assessment test for extra credit.
You don't need a break.
Alright, let me pass this thing out. Let me first shut this sucker down. Pass this assessment test out.